Okay, you can start now. Yes, yes.
Okay. Well, let them move on first.
Watch. One of my knees. They have this mic color. It's fine. You want this one? You stand. That is randomly. You will stand and ठीक है? Yes, start.
You're starting?
Yeah.
Okay. Great. Well, oh, you're leaving. I see. I'm on my own now.
Yeah. No, I'm going to send the link for the lectures.
So thank you, Professor Shukla, and thank you all for being here. So you can hear me okay? Yes. Good morning. Good morning, sir. Oh, that's good. Better than my student at Columbia. Stay away from here a little bit. So I guess it's a pleasure for me to be here. Second time in India. First time in Delhi. First time in Varanasi. First time in Agra on Saturday. So beautiful. And maybe a little bit of myself. So the topic of structural biology and drug design. So I'm a structural biologist by training. My undergraduate study on small molecule crystallography. My PhD on protein crystallography. My postdoc's still on protein crystallography. And I spent 5 years working in a pharmaceutical company, a German company in the US, so doing structure-based drug design. So I have a lot of interest in small molecules, how they interact with their binding target proteins or RNA or DNA for that matter. So that's sort of also because my undergraduate was in chemistry. So I have a lot of understanding of chemistry, organic chemistry especially. That's what you need for biochemistry, understanding organic chemistry. So all of these come together for my interest in small molecule drug and target interactions. So that's what the plan. I assume that's what you're interested, that's why you're here. But if you're not, please let me know. If you're all not interested, let me know. I can change the course. We'll see how it goes. But anyway, I hope that in the next 5 days, we'll learn something. Or you learn something from me, maybe I'll learn something from you as well. And please ask me questions if you're not sure. But if you do ask questions, please just speak up and speak slowly. I'm sorry, I'm still trying to understand. Anyway, so let's make it a dialogue, informal. So sitting in the front is okay, I won't eat you, that's fine. I had breakfast already, so I'm good. Did I understand? Yes. Not a good joke, I'm live, sorry, I cannot say that. Okay, so the overall plan is today we'll spend talking about the experimental technique of structural biology. primarily crystallography and cryo-EM. So I'll spend maybe the next 2 or 3 hours talking about the theory about it, and then after lunch, in the last hour or so, I'll show you example how you solve a structure by crystallography and how you solve a structure by cryo-EM. Only in 1 hour, do 2 structures. Does that sound interesting? Okay, so how many have done structural biology already? Not you. How many else? A little bit, okay. Biochemistry? Okay, good. Proteins? I see. That's why you're sitting in the back. No response in the back. Anyway, okay. Well, as I said, please ask questions, and then we'll see how everything goes, okay? So let's start on so I have a lot of slides. We'll just go through them and stop me with questions. So structural biology, the basic idea is use structure to understand function. So the first step, of course, is determine the structure by experimental, I forgot to ask for laser pointer. Do we have it? That's your pointer. So, I don't have a mic. So, can you hear me okay standing here? Yes. In the back? No? You okay? No? All right, I'll stand here. So I guess okay, we're getting a laser pointer. So the idea is to use experimental technologies, crystallography, or cryo-EM to determine the structure, and use structure, the second bullet point, using structure to understand function. And the third point is using structure to design better compounds or inhibitors, whatever it is, in a pharmaceutical or in an academic setting, structure-based drug design, essentially. So, structural biology used to be a very difficult field, but then over the last 10, 20 years, tremendous improvement in technology has really empowered this technique, especially computers. You all know CPUs, GPUs, I guess playing games, you all know about GPUs. Huge amount of memory, huge amount of disk storage, and computer graphics are getting much better. And there's something called synchrotrons for crystallography, which I'll explain briefly later. Thank you. So synchrotrons, and then detectors for X-ray photons, and also electrons for cryo-EM. That advancement in detectors is what enabled cryo-EM to be so powerful over the last 10 years or so. I'll explain. And then, of course, robotics, everything now computer-driven, robotics. And then genetic engineering, doing protein expression, all of these, of course, these have a much longer history now, maybe 30, 40 years now, but still. So over the past 10 years, so I think computers, detectors are the major driving force. So as a result of that, structural biology is becoming a tool that almost anybody can do it. And that's sort of the trend now. Most labs don't do structural biology sort of on the side. And I've been teaching at Columbia since 1999. So I showed this sentence first time I taught the course on structural biology, is that structural biology may no longer be a separate field in the near future in the sense that it's only a tool. Just like biochemistry or small molecule crystallography. Used to be a field on its own in the 1950s or 60s. Now it's a tool. There's companies you can send your molecule to and they'll get your structure. Everything okay so far? All right. So I already mentioned crystallography, EM. So these are 2 of the 5 techniques that can give you structures at atomic resolution, or give you atomic model of structures. So including the others being NMR, nuclear magnetic resonance, or electron diffraction or fiber diffraction. So the most powerful techniques, of course, are crystallography and EM, or cryo-EM. So that's what we'll focus on today. I won't say anything about these other technologies. So if you look back into history, so the major achievements of structural biology and to help understand biological or biochemical processes are all, actually, this is up to 2011. Well, 14 years ago now. All of these are by crystallography. For example, the prediction of secondary structures, 1951 by Linus Pauling. Of course, the double helix, 1953, Watson and Crick. And the structure of first globular protein, myoglobin, 1960. first RNA molecule, transfer RNA for phenylalanine, 1973, and then a carbon coat virus, 1985, and then first membrane protein, same year, 1985, the nucleosome, the building block of the genomic material, 1997, the ribosome, 2000, and then RNA polymerase, 2001, and then GPCR, G-coupled protein complex, 2011. And then if I keep on going for the last 10 years or so, because cryo-EM now, you get many more dramatic structures. For example, spliceosome, photoreaction center, or many other things that have been done by cryo-EM over the past 10 years or so, which I will not list here. Okay? So the basic idea here is the structure of a protein determined by the sequence. So this is the slide I made back in 1999. Okay? So at that time, actually for many years after that, it's not possible to predict the structure of a protein based on a sequence. Therefore, you have to determine the structure using experimental means, crystallography or cryo-EM. Of course, how many of you heard of AlphaFold? Oh, okay, I'm teaching something. That's good. Thank you. So since 2021, 4 years ago now, using artificial intelligence or machine learning, whatever you call it, so AlphaFold or Rosetta Fold can now be used to fairly reliably predict structures of any protein you're interested in. Does that mean that we are out of job as a structural biologists? No. It only is, again, it's a tool. And I'm an experimentalist, so the prediction, I'll show you some examples later, or one example later. The prediction actually gives you a very good picture of what the protein looks like, but the details may not still be right. So again, you have to get the structure yourself to really prove to yourself that the structure you have is actually what it is. So there are many details may not be correct from the prediction, especially protein-ligand interactions or protein-protein complexes, all of these things. They can be predicted. The question is how real, how correct are they? So we still need to do structures experimentally to confirm whether they are correct or not. Of course, the prediction gives us a lot of insight as to how to do the experiment. So it's a very powerful, very useful tool that we use all the time as structural biologists. So here I'll show you a brief flow chart of how to determine a structure using crystallography. And then EM, well, as you see, so to study a protein, first you have to make it. Most often nowadays, everything is made recombinantly, so you have to subclone it, express it, and purify it. Once you have purified protein, that can go into EM or NMR already. So that's the easy part for EM. But then for crystallography, you had to have crystals, so you had to crystallize protein. So you had to, there's a step of crystallization, and then you have to diffract X-rays from the crystals, so that's middle step, all dealing with crystallography. Especially something called the phase problem, which I'll explain a little bit later. Essentially, the data collection, phasing, and from that you get an electron density map, and get a structure, and use that, as I mentioned, to understand biology. So the idea of structural biology, using structure to understand biology. So in the next, oh, there's a clock there, that's good. Next, I don't know how long it'll take, we'll see. I will just walk through this flow chart one step at a time to give you a flavor. Not the detail, just a general flavor of things. How many of you are familiar with this process? No? Okay. You are, okay. You're as well. Good, 2. Great, thank you. So if I'm wrong, please speak up. Okay, so how do you make proteins? So as I mentioned, nowadays almost everything is done recombinant, from E. coli, bacteria, or insect cells, or even now 293 cells are very popular for expressing proteins, difficult proteins. And then of course for really difficult ones, they have to be isolated from natural sources. For example, spliceosome, ribosome. So these things came from the original source because you cannot get them recombinant. And then we need milligram quantities for crystallography. So it's a huge amount of protein. Of course, you need much less for EM, probably 1,000-fold or less micrograms will be enough for EM. Okay, So that's another advantage of EM. You don't need as much protein, but still the sample has to be good quality. So how do you tell quality? First, purity. You run the SCS gel. Oh, you all know about SCS gels, right? Good. Thank you. So it has to be very pure, 95% pure. Basically a single band, major band on the gel. And then you want to make sure the protein purifier actually is your protein. Sometimes you will have surprises. You're chasing a band of the gel, sometimes you chase the wrong band, and then you purify the wrong protein. You don't want that to happen. And protein should be homogeneous in solution, which I'll explain why that's the case. And then the protein needs to be stable, because to grow a crystal takes a few days to a few weeks. So the protein needs to be stable during that time frame. Otherwise, you can't grow a crystal. And then appropriate concentration, typically 1 to 100 micromole. We typically start with 10 to 20 micromole concentration of the protein. For prior EM, it's about half a micromole. That's what you need to do. So that's what you start with. And then as I already mentioned, milligram quantities for crystallography. So crystallization, something is an art because we don't have any clue how to grow crystals. Well, how to grow crystal of a protein of interest. However, to do crystallization is very easy. I assume you have all used pipetters, right? How to use a pipetter? Yes? I hope so. Anyway, if you know how to do that, you can grow crystals. But because it's art, anybody can do it as well. Some people do the art better, some do it worse. I'm not good at art. I'm okay at growing crystals. So one of the most popular way of growing crystals is something called the hang drop vapor diffusion method. That's shown right here. So you have basically an enclosed little chamber system. And then in the reservoir, you put in your precipitant, which could be salt, or polyethylene glycol is a crowding agent, or organic solvent. Basically, these compounds, or what you call them, reduces the solubility of your protein. And then on this cover slip, you put 1 microliter plus 1 microliter, 1 microliter protein solution plus 1 microliter reservoir solution. And then you flip it upside down, and then these are vacuum grease right here that seal the chamber. So there's no communication between the chamber and the outside, no exchange, essentially. So if you remember your chemistry, or PChem, physical chemistry, because at your start, the concentration of precipitant is lower here than down here in the reservoir. So water vapor will leave the drop and enter the reservoir. Basically, you want to reach equilibrium, right? To reach equilibrium, the concentration of the precipitant needs to go up here. So when it goes up, the protein will start coming out of the solution because it reaches saturation, it will come out of the solution. And most of the time, it comes out of the solution as amorphous precipitates, useless. Okay? But then if you do it right, you have the right condition here, you get crystals. And that's something, as I said, it's an art. We don't really have any prediction so far. Nobody done AI to say, given a protein, how do you crystallize protein? Nobody has been able to do that. So it's a screening process. You screen hundreds, thousands of conditions to find the condition that give you crystals.
So we have to use different precipitant. Different precipitant we have to check.
Yes, you have to screen essentially different combinations and everything. So essentially, these are essentially cocktails. You can make up all kinds of random combinations to sample as much space as possible. So there are commercial companies that sell these screening solutions as kits. And then you buy them and then use a robot. I'll show you a robot to basically set up the experiment for you. And then you have a robot that can look at the images and tell you, okay, you have something going on here, you have nothing going on there. So you can tell you where to focus looking at. If you have 1,000 conditions, you can do it by hand. We did it by hand. 20 years ago, it's only by hand. And then you look at everything under a microscope because I'll just show you later, crystals are very small. So you have to look at everything under a microscope. So it's a lot of work you're students, so you're used to work. Well, I was a student myself, so I did everything by hand when I was old. I am old. So when I was doing this, everything's done by hand. pipetting, microscope, everything done by hand. Okay? So here's the robot. So you can see, essentially, if you screen 1,000 conditions, by hand is doable, very repetitive, boring. So there are robots. This is called the mosquito. Well, this is proboscis of the mosquito that suck up the solution for your protein, suck up a solution for your reservoir, and then deposit onto. That's how you. And then each one is used for one condition. So and this spool here has 15,000 of these little things. You can essentially using this spool, you can send up 15,000 conditions. We typically use about a couple hundred or 500 conditions. So essentially, we can go through this once in a while. This spool here is about $1,500. It's not cheap. Because this is single use. You use it once and then you throw it away. Plasticware. Anyway, so as I said, if you do it right, you can get crystals. So you can change all kinds of different conditions, obviously, all kinds of different parameters. Protein purity, homogeneity, I already mentioned that. Concentration, what kind of precipitant, and what kind of concentration of precipitant. For example, use sodium chloride as a precipitant. Is it 1 molar, 2 molar, 3 molar, 5 molar? We don't know. You have to screen. Find the optimal condition for your protein. What pH you want to use. What temperature you want to use. Typically, we grow everything at 20 degrees, in the incubator, 20 degrees. But then if you're working with thermophilic organism, you may want to go higher because 20 degrees for them too cold. They freeze. So it's not good for them. You may want to go to 37 or something. And then other additives, so essentially, as I mentioned, kits are available. So if you do it right, you get crystals. So for example, this is a crystal of HIV2 protease. And as I mentioned, this picture is taken under a microscope. So across here is probably 0.2, 0.1 millimeter in size. The length is about 1.5 millimeter. So you cannot see it by eye. So you have to look at it under a microscope. This is probably magnified about 40-fold. And then you can see the crystal. And then the other thing you see is crystal has sharp faces and sharp edges. And that reflects, so this microscopic, what you can see under a microscope reflects how the molecules are arranged in the crystal, which I'll show you in a little bit. Okay? So this I call the Washington molecule. Well, I don't know how. Well, anyway. And here you have crystals, pyramids in the sands of Egypt. I guess that you're familiar with. So this is crystal human renin. So tetrahedral crystals, again, very sharp edges, very sharp faces. But then it's not the beauty contest. Any crystal, any shape, if it diffracts X-rays, is fine. So this is what they call the dagger. Another crystal HIV protease. Looks like this is the handle, this is the blade. And that's, even though not beautiful, but it's good enough for crystallography. We actually determine the structure using this crystal. So it's good enough. And then here's another example. Crystals looking beautiful like this, very sharp edges, sharp faces. This is about 1 millimeter across, a gigantic crystal actually. And then this are flowers of a petal, each one quite small. And then so in the end, it's not how we see that's beauty, it's how X-rays see that's beauty. Yeah. So when you shine X-rays onto this crystal, it does not do well. Even though it looks beautiful by eye, but we throw it out because we know these don't do well. We don't use it. And it's one of these petals that we use for our structural studies. So essentially using that petal, we can determine the structure of this protein, which happens to be called metallic enzyme, doesn't really matter. This is how it looks like. So in the next few slides, I'll go from here to a structure on the cover of a journal, well, my only cover structure of my 40-year career. So I had to show it. Okay, so far okay? No? Oh, I guess when you do this, it means yes, okay. I need to learn, yeah, okay, so that's good. Keep on doing that. So as I mentioned, lysozyme. So we'll use that sort of, say that a few times, because lysozyme is the test crystal for crystallography. Because it crystallizes so easily, it diffracts X-ray like crazy. So lysozyme is used to make sure machines are working correctly. So how do you crystallize lysozymes? Well, first you need lysozyme. How do you get lysozyme? You buy from Sigma. Well, Sigma's in India, is that right?
Yeah,
Okay, so you know what that is. You just order it, that's it. You get 100 grams, whatever. You make it, and then you crystallize using something called the batch method, which is different from vapor diffusion. So what happens here, you dissolve lysozyme to make about 100 micromole solution in water, in pure water. And then you make sodium chloride as a precipitant in acidic buffer, pH 4.7. So that determines the pH and also concentration of the precipitant. And then just mix the two 1-to-1. And then you add the last bit slowly until the solution turns milky. Once it turns a little bit milky, you shake a little bit, it dissolves again, probably maybe another drop, and then shake it, dissolve again. That's it, don't push too much. And then you put that, cover it up, put it in the quiet corner, next day come in, you get crystals on its own, okay? And those crystals, as I said, differ actually very well, so actually machine vendors use them as test crystals to make sure that machines are working correctly. So 2016, my former student Christine, who is now at Genentech in San Francisco, so they bought a new machine, so the vendors sent them a lot of crystals, lysozyme crystals to test the quality of the machine. It's not in your printout. Anyway, so she, I guess, had too much time on her hands, so she spent time arranging the crystals into a Christmas card for me. So I got this as a Christmas card at the end of 2015, so for the year 2016. Anyway, so each of these is a lysozyme crystal. Okay, so what are crystals? You've seen crystals now, sharp edges, sharp faces. The reason they have sharp edges, sharp faces, because essentially the crystal is a lattice. Do you know the concept of lattice? Okay. So essentially, in this lattice, the molecules are translation repeated. They're moving, I'll show you examples on the next slide. So they're all the same. The only difference is their position in space. And they're positioned in the lattice, okay? And then the repeat motif is the unit cell. And the rest doesn't really matter too much. And then the crystal is a convolution, that doesn't matter too much either. Let me show you a picture first. So here's a crystal, 2D, okay? So you can see this is your molecule translated along the vertical axis and the horizontal axis and repeats infinitely. Okay? So you can see they're all the same. The molecules are the same, and it's a regular repeats going back in both directions. Okay? So then the unit cell, you can say, okay, so this rectangle will be the unit cell. So that rectangle is repeated over and over again. So essentially, the crystal is made up. You can take the unit cell and stack against itself in 2D, and then you can imagine third dimension will be the same. Will give you three-dimensional crystal. And the content unit cell will be this molecule right here. Okay, so that's the lattice, and then you have a content in the unit cell. The molecule is the content. The lattice is how the molecule are arranged in space. So that's a crystal. And because this regular repeating pattern, you get sharp faces, sharp edges in your crystal. Okay? Is that yes? That's a yes, right? Okay. All right. And crystals, as you saw the pictures, crystals look solid to you, right? They all like table salt you use to cook. But protein crystals are different in the sense that protein crystals, even though they look solid, they're actually full of water. Typical protein crystals are half water. So one of the standard criticism of crystallography is that crystals are solids, and we are not solid. Well, you know what I mean. We have a lot of water in us as well. So biochemistry happens in aqueous solutions, right? So one of the standard criticisms is protein crystals are solid, so it does not reflect what happens in us, because in us, proteins function in aqueous solutions. But as you see here, proteins are 50% water, half water. So many enzymes, actually, when you crystallize them, even in the crystalline state, they are still catalytically active. They can still go over catalysis, which means essentially that even though they are in this crystalline solid state, they can still do the catalysis, do their regular biochemistry. because of this very large solvent content. And because of this, we need to keep our crystals happy to keep water around it. So if you take a protein crystal, you expose to room air for a long time, they will dehydrate, and then it will no longer be a crystal. So way back then, what we did is we put the crystal in the capillary, you see right here, and then we seal both ends using wax. So that way we maintain, and then leave some water around the crystal. That way we maintain the water vapor pressure so it doesn't dehydrate. Okay. So this is another example. 50% is typical. So you have this crystal where the proteins here, here, and here are nothing, solvent. So this crystal actually, 70% of the volume actually is water. So you can go from maybe 30% water to 85% water in the crystal. Still, generally 50% of the volume is occupied by water. So certainly a lot of water-protein interface. So that's why it's still mostly aqueous in that regard, even though it looks like a solid. So that's crystals. And X-rays, you all know about X-rays, yeah, I guess, so I won't spend too much talking about it. But we work with X-rays that are so-called hard X-rays, which are short wavelength, about 1 angstrom wavelength, which is 10 to the minus 8 centimeters. And this is best suited for studying structures because the carbon-carbon single bond is roughly how long? Questions? You're sitting in the front? No, just kidding. Anybody want to venture a guess? What's the bond length for a carbon-carbon single bond? Speak up. That's fine. It's a democracy. We can have a vote. No? Speak up. I can't hear you. All right. Okay. Too many people in the room. 1.5-ish. Is that okay? Good. So essentially, the wavelength is roughly the same as the bond length, the separation between the atoms. So that's why you can use that wave to see the difference in the positions of the atoms. And the other most important thing is X-ray is a wave. So a wave has an amplitude and a phase. So you need two numbers to define one wave. How far it oscillates, and then how it oscillates relative to a fixed reference point. So that's the phase. How far it oscillates is the amplitude, and how the reference point is to give you the phase. So that's represented by, I guess, this representation. So this is the complex number. I is square root of minus 1. It's an imaginary number, essentially. So how do you generate X-rays? So if you go to a doctor's office, they make X-rays using something like this. You have a filament, electrons go through the filament, and then using high voltage, you draw electron off the filament. You have a target, which is either copper or molybdenum. The electron hit the target, and then X-ray come out. That's typically how X-ray generated at the doctor's offices. And we use that technology as well for our research. And then a synchrotron, as I mentioned at the beginning, is something that electrons go around in the ring, a storage ring. And then, so because they're going around in a circle, so they are being subject to centrifugal force. Otherwise, they'll go straight. So you have to apply forces to make them change direction. And so based on classical electromagnetism, when electrons subject to the force, they emit EM, or electromagnetic waves. And then if the force is strong enough, the wave will come out will be the X-ray range. So that's, and the advantage of this is the beam is much stronger than the home source. It could be 10,000, actually now probably a million-fold stronger using something called X-ray lasers, free electron lasers, which are too much technical detail, but anyway, so we use this a lot as well. I'll show you a picture. So this is the source at home. So you have X-ray coming out of this metal tube. I'm not showing the back, you can see that. There's a capillary right here. And then this is the piece of wax right here. And then this is the so-called goniostat that rotate the crystal around in your experiment. So as you come out, hit the crystal, you saw the crystal right here, which is in this capillary. Hit the crystal, diffracts, and then behind this black piece of paper is a recording instrument that records the diffraction pattern. the X-ray photon that get diffracted from crystal are recorded behind a piece of paper. Different detectors are used for that. And then more recently, another way of doing this is using even a smaller, this is the one I showed you earlier, something called an imaging plate. It's a piece of X-ray film that's reusable. And this one is something called the CCD, charge couple device. And then you can essentially record the image directly without having to be scanned again, or this and that. And then here the crystal actually sits, you can see, not in the capillary. It actually sits directly in the X-ray beam, because this is something called cryocrystallography. So the crystal sits in the loop instead, and the loop is frozen at 100 Kelvin, which is liquid nitrogen temperature. So if you look back here, you see this fuzz right here. This nozzle right here is blowing nitrogen gas at 100 Kelvin to keep it cold, to keep the sample cold. And then actually shine on the crystal, and the diffraction part is the same. What's the advantage of that? When you keep the sample this cold, they last much longer in the X-ray beam, because when you shine the X-ray beam onto a crystal, all kinds of chemistry start happening. Free radicals start being made. At room temperature, they'll start destroying the crystal. But if you keep it liquid nitrogen temperature, everything gets slowed down. So decay much slower. So a single crystal can be used for all you need to do for your experiment. You don't have to use many, many crystals, which is a big advantage in that regard. Okay, so that's the home source. And synchrotrons, the one closest to Columbia in New York is the one on Long Island, New York. It's called Brooklyn National Laboratory, National Synchrotron Light Source. And this was in existence for 30-some years, I believe. And then 5, 6 years, well, maybe more than 5, 6 years ago now, maybe 10 years ago now, it got retired. And then a new synchrotron ring right here is built. That's the sequel. Okay, so that's the new ring. The original ring is right here. You can see how small it is compared to this one here. So a lot more things can be done here now that we do use. And back when I was a student, even when I was first faculty, you had to go to the synchrotron store yourself in person to do the experiments. Now, robotics, you don't go there anymore. You just send everything there by FedEx. Send the crystal there by FedEx, and then you control everything using a web interface. And then you can basically examine your crystal at the ring and then do your experiment. But still, the synchrotron runs 24 hours a day. So used to be would go there, like 6 of us would go, and then would take shifts, 6 to 6, 6 a.m. to 6 p.m. So, and then the other group gets 6 p.m. to 6 a.m. That's a lucky group. They work through the night. Same thing here. Even though you work through a web interface, you could still be working at 2 o'clock in the morning. But you'll be at home in your pajamas, possibly. Okay. The detectors, I mentioned the imaging plate, charge couple device CCD, and now the most modern technology of course CMOS, or active pixel sensor display detectors, and that really is the latest technology. And as I mentioned earlier, this technology also is what's critical for cryo-EM. Having this instant readout detectors, this CMOS detector, that's basically what's in your phone. When you take a picture on your phone, you're using the CMOS detector. Okay, so here is an actual diffraction pattern from a crystal. So you see, basically, the pattern contains spots, right? And then the spots have different separations. Some are further apart from each other, some are closer to each other. You see that? And then most importantly, you see spots have different intensities. Some are very bright, some are very weak. So as you see later, the fact you have spots because it's diffraction from a crystal. Crystal is a lattice, and diffraction from a lattice is not a lattice. And that lattice is the spots. And then the different separations represent the unit cells. So the fact you have spots, the fact you have different separations just means you have lattices in your crystal. But most importantly, the structure of your protein, the structure of a molecule is encoded by the intensity of the spots, how bright they are, how weak they are. So that's the concept of the data collection, is basically find out how strong the spots are, how weak the spots are. So you have to go in and measure the intensity of each of these spots. So that's the concept of data collection. But jumping ahead a little bit, so what can you say in general about X-ray diffraction? First, only electrons diffract X-rays. You know molecules made of electrons and nuclei, but nuclei we do not see by X-ray crystallography. So we only see electrons. That's why by X-ray diffraction, we talk about electron density, because that's what we see. So you hear about electron density all the time because we see electrons. But first, when you see electrons, the nuclei cannot be far away from the electron anyway, so it's not a problem. But that's because electrons are much lighter than nuclei, that's why we can see them. And the diffraction actually form an order pattern. I already explained that. Essentially, you have a lattice, form a crystal. Then you shine X-ray on it, you get another lattice. And then diffracting power is proportional to atom number. So the more electrons you have for that element, the stronger the diffraction. So as a result, hydrogen, which has only one electron, has very weak diffraction in X-ray crystallography. So that's why oftentimes we don't really include hydrogens because we can't really observe them directly. Okay. And then the larger the crystal, the stronger diffraction, which sort of makes sense. The more sample you have in the beam, the stronger the diffraction is. And then the larger the molecule, the weaker the diffraction, which I think makes sense as well. So for example, you're working on ribosomes, very large molecules. Those crystals do not diffract using a home source. You have to take them to a synchrotron using a much stronger source so that you can see diffraction. Okay. And then when you shine X-ray onto a crystal, you're shining the entire crystal, which means everything in that space is averaged out. So X-ray diffraction is a space average over the entire crystal that's exposed to the X-ray beam. Also, it's a time average because essentially when you collect data, when you collect your diffraction data, it takes seconds. It used to take hours, now it takes seconds. But still, during that second time frame, everything is averaged out. If you're looking for faster processes, microsecond, millisecond processes, or picosecond processes, you have to do, there are technology to do that, but not traditional technology. You can't do time-resolved crystallography if you're using special setups. Okay, but typically, crystallography is a space and time average. So here's another example. So here you have one molecule like this, and this is all calculated, okay? So you calculate the diffraction pattern for that molecule, it looks like this, very, very complicated. Also very, very weak, which you cannot see here on this slide, unfortunately. So very weak signal, very complex signal. Now when you have two molecules, you can already see the pattern changes. And we have basically here 3 by 5 little small lattice. You already see in spots. Because essentially the diffraction from each of the 15 molecules here are interfering with each other. When they interfere, sometimes they add up, sometimes they subtract. And then only at certain points they add up, you give a spot, and then other places you get nothing because they're subtracting out. Because you have a lattice here, you get another lattice. So having a crystal give you a lattice, which is simplify the diffraction process. The other important point is this amplified process, because even though this looks the same color scheme as that, this signal is 15 times the signal here. And then in the typical X-ray diffraction experiment, you have probably 1 billion molecules in the beam. That means the signal is amplified about 1 billion-fold. So without that crystal, you're not going to see the diffraction unless you use a very, very strong X-ray source, which is free electron laser. So that's, using a free electron laser, you can use very small crystals or even single molecules, in theory. All right, so another important concept we should talk about is something called the Bragg equation. So the idea is when you think about X-ray diffraction, so William Bragg back in the early 1900s sort of thought about this as reflecting. When you look at the mirror, you're seeing your own reflection, right? So he was thinking about X-ray diffraction as a reflecting of X-rays. So you can see these are so-called lattice planes. Let me show you an example of that. So here, crystal again, you have planes of molecule here, this way as well, and also many different ways you have lattice planes, right? You can draw planes in many, many different ways. So what he thought is these are two lattice planes. You can actually coming in at angle theta to this plane, and then actually going out as a reflection. Now you have not actually coming in, hitting the next lattice plane, and then going out. Okay? So these two waves are parallel to each other, and they will interfere with each other. But this wave travels a little bit longer than this wave. And the longer distance traveled based on trigonometry is 2d sine theta, assuming d is the distance between two planes. This is theta, which means this angle is theta. That's d. That means this is d sine theta, so twice of that. Do you see my derivation? Okay, homework. Anyway, so that means this wave traveled a little bit further than this wave by 2d sine theta. And the only way the two waves can add to each other as interference is this distance is a multiple of the wavelength of the wave, so that they all oscillate the same way. So that's how he derived this equation. That's the Bragg equation, 2d sine theta equals lambda. So this is not like regular you see anywhere you stand, you see a reflection. Here, you only see a reflection when theta satisfies this equation. Otherwise, you see nothing. So again, that explains why you have spots. Because for most data doesn't satisfy the equation, you don't get anything. So you get blank space. Only when theta satisfies the equation, you get a spot coming out. And because this is the way he thought about this, so that's why the diffracted spots are often called reflections, reflecting from some lattice plane in your crystal. So theta is defined to be the angle between the X-ray wave and the plane. But as you see, the plane can be anywhere in the crystal. You have no idea where it is. So how do you know theta experimentally? So the way to do that is if you look at the beam that goes straight through the crystal. Okay? So this is the beam that goes through the crystal. This angle is theta by trigonometry. This angle is also theta. So that means the angle between the beam that goes through the crystal and the diffracted beam is 2 theta. Right? And that you know very easily because that you can do by trigonometry. Because you know where the direct beam hit your detector, you know the diffracted beam where it hit your detector, and then the rest is trigonometry. I'll show you that in the next slide or in a couple slides. That's actually an important concept we'll talk about as well. Okay. So it's a general concept. So the X-ray diffraction is a physical process. Mathematically, it's a Fourier transform that's shown right here. So here's your crystal, and then you do a Fourier transform of your crystal, you get the diffraction pattern right here. Okay? And then this molecular transform is the content of the unit cell, and that gives you the structural factor that encodes the structural information. I know this is probably too much for you, but that's okay. I'll show you maybe an easier understanding on a later slide. So here is another example of a diffraction pattern from a crystal, from a RAS protein. So you can see again spots, different intensities. Okay? And I won't go through symmetry and other stuff. But one thing I will mention, because this, again, it's a lattice. So every spot on this lattice can be referred to by its coordinates, which will be integers. For example, this spot right here will be 1 along the A star axis, and then this spot right here, 1 on A star, 1, 2, 3, 4, 5, 6, 7, 8, 8 along B star. So each spot on this image can be represented by 2 integers being its coordinates. You know about XYZ coordinates in the Cartesian system, right? But those are real numbers. because we're dealing with the lattice here, so all the coordinates, every spot here has an integer coordinate. That's called the index of the spot, which is written as HKL in diffraction theory. Okay, so each spot in your diffraction pattern has an index, which is 3 integers, HK and L. And the rest I will skip over, that's okay. So the concept of X-ray diffraction data collection essentially now is for each reflection, we know its index. We need to measure its intensity, also the error in that measurement, intensity and sigma in the measurement. Okay? And then when you do this measurement, sometimes you measure same reflection several different times. You can then determine how well they agree with each other, how well their measurements agree with each other. And that's used the concept called R-merge. That's which I'll explain. So first let me show you how it works. So here again is another diffraction pattern. If you see this part here, zoomed in over here, spots, different intensities, right? So first we find out the spots. I'll show you in the afternoon, hopefully for lysozyme, how it works. But this is just briefly, we find out the spots. Then we can use that, determine the unit cell parameters, everything else. We can predict where the spots are. Then you go in, predict where they are. Then you can see each spot predicted. And then they are enclosed here, so you can go in now calculate intensity for each of these spots. Also, by you're doing the prediction, you know the index of the spot as well. So for example, this spot has some index, you can calculate intensity. This one, the next spot here, you can see this one is much darker, so stronger than this one. So it may seem to be a very complex process to do manually, which is true. For a computer, this is a piece of cake. It takes less than a second to do for a computer. So R-merge essentially is if you have more than one measurement for each reflection, R-merge is a measurement of how precise, how well they agree with each other as a percentage values. Typically 5 to 10% is what we observe in our data set. And then more recently, another concept is called CC1/2, correlation coefficient 1/2. 1/2 means you split the data in 2 halves. And you process data from each half individually, and then you compare them to see how well they correlate to each other. You calculate correlation coefficient. So if they're fully correlated, it will be 1. If they're not correlated, it will be 0. So essentially, you want this number to be close to typically around 0.3, 0.5 or so. So mostly correlated. That means that's where you still have information, you still have signal. Okay. So this is typically what the data collection looks like. HKL index, intensity, sigma. So, and then it goes on and on. And so this data set has 133,000 reflections. Okay. So for a human to do, very difficult. For a computer to do, a couple minutes. Now we have HKL intensity and sigma. Sigma being the error of that measurement. So what we measure is the intensity. Intensity is actually proportional to the amplitude of the X-ray wave, because each spot is an X-ray wave, a diffracted X-ray wave. And that wave has the amplitude and the phase. And the amplitude squared is the intensity. So what we're measuring is the square of the amplitude of the wave, how many photons are there in that wave, essentially. And that amplitude is related to the structure of the molecule by this structure factor equation right here, the fundamental equation of crystallography. So XYZ define the position of an atom in the unit cell. HKL, I already told you, is the index for that reflection. Then B is something called the temperature factor, measures thermal motion of the atom in the unit cell. Sine theta over lambda, came from Bragg equation. So that has something to do with basically how far the reflection is. So you can see that essentially every spot in your diffraction pattern come from every atom in your crystal. summation goes over all the atoms in your crystal. So every spot, every atom contributes to every spot. So you cannot say, okay, that spot in my pattern came from this atom. Doesn't work that way. It came from all the atoms in the crystal. So this is another way of looking at Bragg equation. As I said, you actually coming in, hit the crystal, the direct actually go hit the detector right here, and the diffractive beam hit it right here. So I just showed you earlier for the Bragg equation, this angle, the angle between the incoming direct beam and the diffractive beam is 2 theta, right here. Okay? So, and we know the distance between the crystal and the detector because that's what we did the experiment. We can measure on the detector the distance between the direct beam and the diffractive beam. So then this is basically, typically this angle is 90 degrees, the right angle. So trigonometry, that means 2 theta is arctangent I over d, right? Which means then from that you can calculate d, the separation between planes in your crystal. And this d is related to an important concept called resolution, how much detail you can see in your structure. So the higher the resolution, the smaller d, because smaller detail you can see. Okay, so 10 angstrom resolution is very bad. 3 angstrom, that's what typically we'll work at. And 1 angstrom will be very, very good. Now you can resolve things that are about 1 angstrom apart. Okay, so you can see then to get to higher resolution, which means you need smaller d. To get smaller d, you need larger theta, which means that larger 2 theta means the spot needs to go further out, away from where the direct beam hit the detector. So first thing we look at when we look at the diffraction pattern is how far the spots go. If spots go very far away from the direct beam position, that means crystal diffract well. If they don't, that means crystal is not diffracting well. So remember that I showed you a beautiful big crystal of 1 millimeter crystal? When we put that crystal in the X-ray beam, you see spots only around here. We say, okay, garbage, throw it away. And when we put the other small crystal, we put it in, it diffracts all the way to the edge. That means it's higher resolution. So essentially that's how we tell how good our quality is, by X-ray diffraction, by how far the diffraction goes away from the direct beam. The higher it goes away, the higher resolution. Okay? So what's the concept? Let me show you examples. Resolution is a critical concept, so let me show you some examples here. So here's the alpha helix. The red drawing is an alpha helix. Okay? So at 5 angstrom resolution, the simulated electron densities in gray. Can you see that in the back? You can see it okay? The electron density? Yeah? Okay. So essentially at 5 angstrom resolution, the alpha helix is shown basically as a sausage. You can't see any detail. You do see a sausage that reflects the general shape of the helix, but you don't have any detail. Then at 3 angstrom resolution, you can see now the main chain of the helix winding around. And then some side chains start popping off the helix. So you sort of see some details now. Then at 1.5 angstrom resolution, you see even more detail. You can see now here, you have a very nice bump here for the carbonyl of the main chain where you don't see that 3 angstrom. At 1.5, now you can see carbonyl. You can see in sort of individual atoms along the main chain now. Carbonyl oxygens now sitting in their own little spherical density. So you can sort of see the difference between 5 angstrom and 1.5. So for most structures are between 3 and 1.5 angstrom resolution. So we do see a good amount of detail about your protein or nucleic acids or whatever it is.
So if we are getting resolution of 5 angstrom, so we have to again go for recrystallization.
Sorry.
If we are getting resolution of 5 angstrom in a crystal, so that means the crystal is not good.
That means the distance between. Yes. So 5 angstrom resolution means features about 5 angstrom you can resolve. You can see at different peaks in the electron density. So let me show you another example here. So here's actually from my own research at the company. So here's the phosphotyrosine. Okay, at 1.8 angstrom resolution. So red again is this model, and then in blue or light blue color is electron density. So around the phenyl ring of the phosphotyrosine, you can see we have a donut, right? There's a hole in the middle, because across the ring is about 2.2 angstroms. So they are resolved as separate features. But then the two carbons in the ring, this is about 1.3 angstrom. So they are not resolved. Does that answer? So do you understand the concept of resolution? So essentially, at 1.8, you can resolve things about maybe 2.2 angstrom away. You can sort of see a hole in the middle because they are being resolved as separate features. Okay? I'm sorry. Is that what?
3D splicing, in which you cut the structures in like 1 hour or something. You split the structure and then do it separately.
You mean separate it?
Yeah.
Separate what?
You're saying that like the angstrom is 3, so you will build it at separate.
Well, let me show you another example to maybe that answer your question. So this is 1.8, and this crystal actually turned out to be very, very good. So I collected data to 1 angstrom resolution. So this is what it looks like 1 angstrom resolution. Did that answer your question? Okay, that was my guess. So you can now see, even within the ring, each atom now is a separate sphere. They are being resolved with separate features, right? And then the phosphate, you can see phosphorus, oxygen, they're completely separate from each other. Okay? So obviously, this one has more information than this one. Here you can see individual atoms. Here you know it should be a phenyl ring, so build a phenyl ring underneath it. So this is illustrate the power of resolution. But then you say, what's the big deal? This is 1.8, this is 1, less than a factor 2. In biology, factor 2 means nothing. Well, I'm on camera, so I'll be careful. So why is that? Because we are dealing with a 3-dimensional process. So resolution is a 1-dimensional number, but then in the end, you need to make it 3-dimensional, cube it, which means then a 2-fold difference in resolution means 8-fold difference in number of reflections you can get. So I'm showing one example right here. So this protein, whatever it is, at 3 angstrom resolution, it has 7,000 reflections. At 2 angstrom, 24,000. At 1 angstrom will be 27-fold of what at 3 angstrom, so 200,000 reflections. So the reason you can see more detail is because you have more experimental observations. That allows you to see the detail. So now the question is, why don't we all collect data to 1 angstrom resolution? Because it's so powerful to see so much detail. Well, mostly because your crystal is not good enough. Crystals is all, I guess you can say at the beginning when I show you crystals, to me it's always magical when I see a protein crystal. Because I don't have time to go through the math here. A protein crystal typically contains about a billion, a trillion molecules in that crystal. And you saw the example. In the crystal, all the molecules need to be the same. So you're talking about a billion molecules, or maybe a trillion molecules lined up perfectly in the crystal. To me, that's magical. And of course, as you can imagine, it doesn't always happen perfectly. So you have defects. When you have defects, you reduce the crystal quality. So that limits how far the crystal will diffract. So that is the most limiting factor as to how far your crystal will diffract, what kind of resolution your structure will be at. So crystal defect is the most important point. Crystal size is an important problem, but now the actual source is getting stronger and stronger that can compensate for smaller crystals. And then technical limitations is really no longer an issue. beam intensity, detector sensitivity, this and that. Technology nowadays, this typically is no longer the issue. So we do the best we can for your crystal using a synchrotron radiation to go to the high resolution possible. So that's all what we do. And to really look at how far your crystal diffracts, you look at the R merge as a function of resolution. So here's resolution, 50 angstrom down to 2.4 angstrom resolution in shells. And then you look at the R merge. So at low resolution, strong reflections, high intensity, very low R merge, 6% or so, which actually is on the high side. On the good crystal, this could be like 2%. And then the high shell is about 30%, which is still pretty good. And then on the right here, you have the column CC1/2. Remember, that showed you to divide the data in two halves, and you calculate correlation coefficient. You can see a low resolution correlation coefficient 0.99. They are essentially the same. So there's definitely signal there. In this case, even at 2.4 resolution, the CC1/2 is 0.8. Still very good. So based on this, you could say this data set probably could go a little bit higher, maybe 2.3, 2.2, but somewhere around there. So this is what we use to tell us what resolution our crystal diffracts to using these numbers. Unfortunately, crystallography is a lot of numbers. That's what you're seeing here. But if you know, once you know what to look for, you can ignore most of the other things. Just zoom in on the ones, most important number, which is this number here and this number here. They'll tell you what resolution you can get to. Okay, so it's an hour now, is that right? Okay, phase problem. So I mentioned X-rays are waves. So I spent a lot of time talking about data collection to give you intensity. I said nothing about phase. Remember, wave needs amplitude and phase. I said everything about the amplitude. I said nothing about phase because we have a phase problem, because we cannot measure the phase. Because X-rays are such short wavelength, which means high energy, there's nothing can focus them. Like if you look at the light microscope, you have lenses that can focus the light to get your image. But X-rays, there is no lens that can focus them. So you lose the phase information. So we have to generate the phase information indirectly. So this is an example of a lens right here. You have an object, go to the lens will focus the waves, the light rays, get you an image. Either smaller or larger, doesn't matter. At least you have a lens here that can give you the image. And for X-rays, we cannot do that. We do not have a lens here. So we replace the lens with a computer. We use a computer to focus the X-ray beam to get you the image. Okay, I'll show you how to do that. That's basically doing the inverse Fourier transform. So here is your diffraction pattern. You do the inverse Fourier transform, the negative sign here. So once you know the X-ray wave, you can calculate the electron density at every position in the unit cell. X, Y, Z being any position in the cell, and this being a diffraction pattern. And then you can use this inverse Fourier transform to calculate the electron density at every position. And that is the diffracted X-ray wave, which has an amplitude and the phase. Amplitude, as I mentioned, is proportional to the square root of intensity you can measure experimentally. So this you can determine experimentally. This we will try to determine experimentally using, we cannot measure it, we cannot measure it directly, but we can determine it indirectly. Once you have this information, that information, you can plug into this inverse Fourier transform, you get your electron density everywhere in your crystal. So that's the basic idea. So right here, so we're done with this part. So how do you do the phasing? And this part actually technically is quite involved, so I will not go into any of that. I will only show you a couple of slides, just give you the general concept. So the classical way is something called the multiple isomorphous replacement. A mouthful, sorry about that. The basic idea is you introduce heavy atom compounds into your crystal. So how it works, do you remember inorganic chemistry? So if you have a cysteine side chain, which has a sulfur in the side chain, sulfur like to bind to mercury or platinum. So if you introduce a mercury compound into your crystal, that mercury compound may bind to a cysteine side chain in your protein and be positioned correctly there. And then once you do that, you can then collect data on that derivatized crystal, and then by comparing the data, you can determine the position of the heavy atom, the mercury compound. Okay? Why mercury? Remember, diffraction is proportional to the number of electrons in the element. So protein atom, typically carbon, nitrogen, oxygen, has about 6 or 7 electrons. Mercury has 80. So many more electrons, much stronger diffraction, a huge spike in your signal. That's how you can find it. If you put in another carbon, good luck finding it. You're not going to find it. So you have to put in something that's different. In this case, something very heavy, many, many electrons. Mercury, platinum, gold, uranium, lanthanides, all of these heavy elements, many electrons. Now you have a chance of finding them. Once you find the heavy atom compounds, you can then use that bootstrap and get the phase information for your protein. So that's the basic idea. Second method is so-called matte phasing, selenium-methanol matte phasing. And in this case, the technology is, proteins contain methionines, right? Methionine also has a sulfur in the side chain, but it's methylated. So in this case, rather than using methionine, we replace methionine with selenium-methionine. The sulfur replaced by selenium, which is one element down, one row down in the periodic table. And selenium, because a little bit heavier, again, it has unique property different from all the other atoms in your protein. And that property is something called anomalous scattering, which I will not have time to describe. But anyway, using this different unique property of the selenium atom, again, we can locate that selenium atom in your crystal. And from that, we can determine the phase of your protein. So these are two. The multiple isomorphic replacement was the original method, the first method that was used to solve protein structures, to many, many years developed, and still used today, but rarely. And then selenium-methanol method was developed by Wayne Hendrickson, actually at Columbia, and used for many years as well, because much easier to do than soaking heavy atoms. But now also not used as much anymore. I remember actually when the last selenium structure was solved in my lab, probably 7 years ago now. So nowadays, the more popular method is something called molecular replacement. So this idea is actually quite simple. So the idea essentially is you want to work on your protein, but then there is already another protein somebody else did already. You know what your protein looks like, what you don't know is how that protein sits in your crystal. So you can use the structural information from somebody else's work and to determine the structure of your protein. So you don't require any selenium, don't require any heavy atom. You can use direct calculation to give you the structure. But now you say, okay, what I'm working on actually had no model, no structure, nobody else worked on it. Well, that used to be a problem. But then there's AlphaFold now. So essentially, almost anything you want to work on, you have a model now from AlphaFold or Rosetta Fold. And that model oftentimes is sufficient to determine the structure by molecular replacement. So that will give you a starting point to give you the phase information and determine your structure. So that's why now, even in my lab, when we have something new, we just go to AlphaFold or Rosetta Fold, get the model, and use that as a starting point. And most times it succeeds getting the structure. So then that's the first we tried. It takes only a few hours, a few days, rather than having to spend time make selenium meth protein, all that stuff. A lot of extra experiment you have to do. This is all done on the computer, very cheap, doesn't cost anything. Okay, so now we have the phase, so we can calculate the electron density, as shown right here. So here is the example of the electron density. So what is done here is unicell is three-dimensional. You cut it up into slices. You plot the electron density on each slice as the mountain ranges. okay, contour plots, like geology or geography. And then you put them onto a transparency, and then you stack the transparencies up. So essentially you can see the stacking right here. So these are different layers of the transparency. So now it gives you a sense of the overall shape of the protein. So where you have a lot of contours is where the electrons are, electron dense region, that's where your protein is. And here you can see the solvent channels. Remember, crystal is 50% solvent? That's what you see right here. So the protein is right here, and solvent is right here. So now your protein is here. So the next part is you have to interpret the electron density you see here based on your protein. It tells you your protein here, tells you nothing else. Doesn't tell you where the N terminus of the protein is, doesn't tell you where the C terminus is, doesn't tell you where residual tyrosine 31 is. You have to go in and interpret it manually. Used to be manually. Now, again, you can do it automatically now. So the first thing we look for is secondary structures. So what are the secondary structures in the protein? Alpha helices, beta strands. So you look for those. So here you have a beta strand right here. Extended structure going across the page, that's a beta strand. So a single beta strand is not happy. You want to be in a beta sheet. So then you have this beta strand next to it, second strand, third strand, fourth strand, fifth strand, sixth strand. Don't believe me? Well, okay, you don't have to. But you can sort of see this strand is in the page or the screen. This strand's going into the screen already. So it's a 90-degree twist in the strands as you go around six-stranded sheet. That happens very often in protein structure. They are not regular. There are a lot of twisting and turning going on. Okay? And over here, this big blob, dark blob right here is the alpha helix. You're looking down the helical axis. So here you see all the main chain. So now, on the next slide, I'll show you this, the electron density of this part looking from the side. That's shown right here. So on the previous slide, we're looking from the top down. Now we're looking from the side. So here you see the alpha helix. This is the main chain. Here the side chain popping off. So the electron density doesn't tell you where the residues are. It doesn't even tell you which way the chain is going. Which way is the N terminus, which way the N terminus is, which is C terminus. So here you have alpha helix. So N terminus at the top or the bottom? Do you know? Well, in the case of helix, actually, we do know. Because, let me show you a model. So here's the helix, right here. The side chains are the little blobs, the pink blobs. You can see the side chain all point up, right? And in this model here, N terminus at the top, because this is essentially N, C alpha, C O, right here. So N terminus at the top, C terminus at the bottom. So in the helix, the side chains points towards the N terminus, right? So that means that in this case, you can see all the side chains point up. That means the top is the N terminus, bottom is C terminus. So in the case of helix, you can sort of tell which way the chain is going, N terminus at the top, C terminus at the bottom, and then by looking at the shape, the side chains, you can start interpreting which residue it could be. For example, something like this, probably the phenylalanine, because round, globular structure is probably phenylalanine. So you can match that to the actual sequence of protein. Now you have to interpret this piece of electron density based on the actual sequence of your protein shown in red here. You can also build in a model for your sequence. So you have phenylalanine, tyrosine, arginines, two arginines right here. So here's the main chain, here are the side chains popping out. So now you have the electron density determined using X-ray diffraction. And now you have a model built into the electron density that satisfies the electron density, also matches the sequence of your protein. So you have interpreted this piece of electron density. So you have a model now for your structure. So you do the rest, do the same for the rest. So here is the six-stranded beta sheet. And this protein often actually turns out it binds GDP as well. So we locate the electron density of the GDP in the structure as well. So we have a model. Next part is refinement. Refinement means to minimize the difference between the model and your experimental data, which I guess you all know the concept. To You adjust the model to minimize your experimental observation, the difference. So that's done using a least square process. So you take the observed structure factor, the square root of the intensity that you observed, and then you can calculate based on your structure right here, each atom, temperature factor, and then for each atom. So you can adjust these values to minimize this difference, the least square approach. Okay, so that's the refinement. And the way to monitor this refinement is something called another R factor, the crystallographic R factor, which is the difference between the observed value and the calculated value as a percentage. And typically for protein structures, this R factor should be about 20% or so, 15 to 20%, somewhere around there. Okay, small molecule actually is much more accurate. R can be down to 2 to 5%, very accurate. Actually, crystallography is a very accurate science. If you do your experiment carefully, you can get very good agreement between your observed data and your model. Very, very good agreement. And then when you read papers, you also have something called the free R factor, which I guess you all heard about cross-validation. You know that concept in statistics? So, okay, let me try it. So the idea is you have so many reflections, right, that you use to do the least squares. But then this whole process can be biased. So the idea that you leave 5%, 10% of those reflections out. You don't use them. You only refine using 95%, 90% data to minimize the difference between those. And then you see how the remaining 5%, 10% are doing. If you go in the right direction, the remaining reflections will also start agreeing, right? If you don't, if you're not doing the right thing, they will not care. They will stay very different from your model. So this is a way, so that's why it's called free from the refinement process. So it's a free R factor. And this will give you an independent way of measuring or assessing how well your refinement is going. Got it? Okay. Let me drink some water, celebrate that. In reality, well, I had to show this because I need to explain this later. So, as you saw, well, let me put it this way. So in reality, what we are minimizing, not only the crystallographic term, the difference between observed and calculated structure factors, we also minimize the difference between the structure model and the ideal model. Because we're dealing with proteins and nucleic acids, we know what they look like. We know amino acids look like that because there are a lot of short molecule structure of alanines, tryptophans. We know the bond length, bond angles of these, all of these building blocks. So if you get the protein structure that look very, very different from these small molecule structures, very likely that that model will be wrong. So we basically restrain the bond length, bond angles, torsion angles, planar groups, chiral atoms, because C alphas are chiral. So if you refine a structure with a C alpha as planar, well, I guess you'll know that's wrong, right? So you want to restrain this so C alpha is tetrahedral. I also write chirality, not L, not D. All of these. So we include all of these restraints in our refinement so that the resulting model will make sense in terms of chemistry or biochemistry. For example, phenol ring I showed you earlier is planar. So if you refine the structure but the phenol ring start buckling, that would not be good, right? So that's planar groups. So they are restrained as well. So that makes the model make more sense in terms of chemistry and the biochemistry of the protein and the nucleic acids. So that's included as well, and that's something you will see reported in the papers. So that's shown right here. So I guess you have all, maybe I should not say them. You have read structural biology papers, is that right? Have you seen them? No? Okay. Let me show you. So in most structural biology papers, there is something called Table 1. Well, by chance, that's the first table we show because that's the table of statistics from either crystallography or cryo-EM. Statistics on our data quality or our model quality. So this is sort of the way you tell. So here is a table. It has 3 structures. And then each row has a unique information. So here's this row here, which protein is in that crystal. Skip space group, doesn't matter. Resolution, it tells you this crystal goes to 2.3 angstrom resolution. This one's 3.0, this one's 3.3. So that's the best we can do. You can also see this one actually complex between 2 proteins. So that's the best crystal can grow. Best data we collect, 3.3. And then our merge, as I explained earlier. So overall, 5%, very good. 10%, this will be worse. This is 5%. And then at higher resolution shell right here, that defines what resolution can go to. Still, it's only about 20%, 30% or so. So we probably can push a little bit higher, but I think we're being a little bit conservative here. But it's a good number. 2.3 angstrom, 3 angstrom, 3.3. And then here, R factor, that's for the model. Agreement between the model and the observed data, 20% overall. Okay, and then free R factor is about 6% higher, and that's typically what we see. Or what we'd like to see, the free R factor should be about 6% or less compared to the R factor. They should not be too divergent from each other. If they're very different from each other, that means something probably is wrong. So when you read your papers, you should also check on that. And then RMS deviation in bond lengths, how well your model agree with a theoretical ideal model. Typically 0.01 angstrom is what we like to see. 0.015 is fine as well. And this is between 1 and 2 degrees. This is bond length, bond angles. Okay, so 0.01 in bond length, 1 degree or so, 1 to 2 degrees for bond angles. So this is something we report for all papers. You see that in all papers, essentially a table like that. They give you a sense of the quality of the diffraction data and the quality of your model. Okay? And then different journals have different formats, but general concept is still the same. So this one, 2.9 angstroms, 2.9, 2 different structures. And then R factor, free R factor. Our work is the working R factor or the general R factor, 6% difference. Here is about 3.5% difference. Okay. So, and then RMS bond length, bond angles. So that's sort of what you look for. So we'll take a break at another 10, 15 minutes. We'll take a break in 10, 15 minutes. Okay. We're almost done with the crystallography part. I'm surprised we're done almost. So when you look at the paper in structural biology, either crystallography or EM, how do you tell the quality of a structure? So these are a few things that came down with. I wrote down, so that can help you to sort of evaluate. Many of you may do work on the protein yourself. You want to say, okay, how good is that structure when you read that paper? So resolution, how high resolution it goes to. As you heard, the higher resolution, the better, but of course that's limited by the quality of the crystal. So don't blame the authors that are going on to 3 angstrom, because that's probably the best they can do after years of effort. That's a major achievement already on its own. So, but check the number, and then how good the data is, our merger of CC1 have, how good the diffraction data is. And then how good the model is, the R factor, and more importantly, the free R factor of the model. And then this I will skip, I didn't explain that. How good the geometry is, that is how well does the model agree with the ideal values, RMS bond length, bond angles, and then Ramachandran plot. You know about Ramachandran plot? Okay, good. I'll show you again later, don't worry. And then these, I put them down, I didn't have time to explain, so I guess we'll leave that be for now. And clashes, something you'll see later is how well the atoms are arranged onto each other. You don't want them to be clashing each other, because that means it's not physically possible when you have a lot of very bad clashes. So that's something that you need to look out as well. So, do you all know about PDB, Protein Data Bank? Great, finally, some response. I'm getting somewhere, I'm almost done. So nowadays, every structure you go to, every entry you go to has this little plot here, right? So do you know what that means? Yes? Maybe? Should I explain? Okay. So depending on the structure, it has different bars. But anyway, for crystal structure, they also always are free, the free R factor. How it compares against the filled bars percentile relative to all crystal structures. The open bar percentile relative to X-ray structure of similar resolution. Probably you should look at the open bar to compare it among your peers rather than compare it against everything, but whatever. So red is bad, blue is good. So you want the structure to be somewhere around the blue side if you can. So this one, R3, actually is very good, 0.23, 23%, very good R3. Class score, which defined to be here, number of close contacts per 1,000 atoms, which includes hydrogens though. So class score here is 4, very good. Typically class score should be below 10. Okay, that's what typically we'll look for, below 10. So I mean 10 clashes per 1,000 atoms. So what the program does is build in all the hydrogens for your structure and then start counting how many close contacts there are. And then divide that by number of atoms it finds. That's give you the class score. Ramachandran outliers, how many outliers of the Ramachandran plot? And that typically should be very low. This one is 0. Well, cannot be lower than 0. So that's obviously very good. Typically 0.5%, less than 0.5%. And then side chain outliers, essentially how the side chain conformation compared to the expected conformation of the side chain. That's something called rotamers, which, have you heard of rotamers? If not, good. I'll show you later what a rotamer is. Okay? And then RSRG just means real space, the quality, the fit to the electron density. So 5% you can see, I mean, all relative to the rest of the structure. So this will be on the red side, but well, still respectable. Okay? So one thing I should warn you though, essentially is when you look at something like this, when you look at structures that are 10 years old or 15 years old, they're all going to be over here. Because 10, 15 years ago, the refinement software is not good enough to take care of all of these things. Or didn't even care about them, didn't know about them, didn't really force those parameters. So those basically doesn't mean those structures are wrong or something. They just happen to be, back then, the best technology available 15 years ago is not good enough compared to what we have now. Okay, so keep that in mind when you look at something that's older. If you look at my structure from 2006, it's over here. So don't throw me out of the room yet. Okay, so I'm just making sure that you treat me well. Okay, so that's that. Okay, so the next part, maybe you're more interested in, this is not theory, this is practice now. So, okay, you guys are still okay? Should we go on, or should we take a break?
We can take break for 15 minutes.
Okay, that's okay with me. All right, so we'll come back to this.
Yeah, okay. You can have tea and then we can start again.
All right, we'll take a break.
And take tea.